Hello, namaste everyone. I'm Dr. Amit here. So, in the last video from our cardiovascular series, we discussed about development of pericardium. Now, in today's video, we'll talk about development of heart. So, let's start our today topic, development of heart from cardiovascular series. So, now first we'll talk about the timeline of development. Okay. So first let's talk timing. Okay, and these dates are very important for exam. So heart formations begins on the 16th day. Okay, sometime it ask in the MCQs questions. Okay, so the heart formations begins on 16th day, and by day 22. Okay, by day 22 it means fourth week. The heart starts begin spontaneously. Okay, so and the third thing is that the heart is the first functional organ to develop in the vertebrate embryo. Okay, now before going on to these three lines, okay, first we'll see a picture and then we'll come here. So let's understand this peak first. Okay, so actually our heart starts with two small tubes, okay, called as endothelial heart tubes, and as development continue, these two tubes, okay, come closer and fuse together, but not completely over everywhere. Okay, these two tubes. Now that two tubes fuse together, okay, and they fuse only at the cranial end. Okay, at cranial end this is the head side, and in the caudal end they remain separated, or we can say they are free initially. Okay, now let's understand what each end does. Okay, so now the cranial end, okay, this end become the outflow part of the heart. Okay, it means blood flow out from here into developing arteries. Okay, and this caudal end, okay, this caudal end form the sinus venosus. Okay, this caudal end form the sinus venosus, meaning the blood flow into the heart from the body. Okay, so simply we can understand that this cranial end will form the outflow part. Okay, and the caudal end will flow the from the sinus venosus, that is inflow part. Now let's understand this picture. Okay, so in starting the heart tube is straight. Okay, but this heart tube actually bends. Okay, it bends and loops. This is called cardiac looping. Okay, and imagine suppose it's a heart pipe. Now twist it into a S shape. Okay, this that actually what happens. Okay, when heart looping is done. Now if you think why heart looping happens, the heart, because the heart tube grow very fast but the space is very limited. Okay, so it bends and fold to fit into the inside the chest. Okay, so this tube actually bend to fit into the chest. Okay, now we'll think about what will be a direction of looping. What is it D loop and this is L loop. Okay, so in which loop actually the heart looping starts. Okay, so the heart loops towards the right side. Okay, that this is D looping. So mostly D looping, this is called dextro looping. Okay, D is the right directions. Okay, this L looping is not the right direction, this is the right directions. Okay, now what goes after looping? Okay, so the cranial part. Okay, this cranial part will bend and move downward. Okay, and to the right. Okay, and the caudal part, okay, this caudal part will form the sinus venosus atrium and moves upward. Okay, and moves upward and to the left. Okay, so now how it will determine the right and left polarity? This is very conceptual question. Okay, so suppose this is D loop over the right and this is L loop in the left. Okay, so the key concept is the embryo is not symmetrical functionally. Okay, so there is one identity gene called PITX2, PITX2. Okay, this PITX2 actually gene identify the left. Okay, and that will helps the in determining that which will be the on the right side and which will be on the left side. Okay, and what happens, suppose normally this straight tube will goes D loop. Okay, but what if L loop occurs? If L loop occurs, then that will be a dextrocardia. Your heart will present on the right side. Normally the heart will present on the left side. So if D loop occur, then heart will present on the left side. But if L looping occur, then the heart will present on the right side. That is called dextrocardia. Now we'll understand these lines. Okay, that the primary heart tubes loop, because we all know that the heart tube is initially straight, and the left and right polarity will be uh, defined by px2 gene, okay, which is we determine the left. Now the primary heart tubes loops, okay. Now if there is defect in left and right dynein, okay, if there is defect in this scene, then dextrocardia will occur, which is seen in Cartesner syndrome, okay. In Cartesner syndrome, the heart will be on the right side, okay. So that is called dextrocardia conditions. Now the two endothelial heart tubes, as I have shown in the figure, the two heart tubes fuse only cranially, okay, and the caudal ends remain free, which is sinus venosus. So the cranial end form truncus arteriosus, that is outflow of blood, and the caudal end, that is sinus venosus, that will form the site of inflow of the blood. And we all know, suppose this is the heart tube, okay. So suppose this is a straight heart tube, then this is truncus arteriosus, bulbus cordis, primitive ventricle atrium, and sinus venosus, okay. So this part will go upside. So this sinus venosus will come here, primitive atrium. While looping, this part will go upside and this part will come downside. So that's why I have written sinus venosus, primitive atrium, primitive ventricle, bulbus cordis, and truncus arteriosus. Now in the next video, we will talk about these septations of atrial chamber and patent foramen ovale. So if you like this video, please subscribe my YouTube channel and share this video with your fellow medicals. Thank you for listening.